Guys, thanks a million. You're all very welcome to our second medical science graduate evening. Thanks a million. Thanks to our speakers for taking the time out. I hope you enjoy. I'm not going to spend much time introducing anybody because that's what they're going to talk about. Just if there are any questions at the end, you can ask. If you are too shy to ask, we have tea and coffee in the foyer afterwards, and the speakers are going to hang around, so you can ask questions then. Or we'll have a little discussion maybe at the end as well. If we'll see how it goes, okay? So we're going to kick off with Siobhan. So. Okay. Second now, Siobhan. Slideshow from the beginning.
You just right back to the next one?
Yeah, or you can narrow down.
Yeah, perfect.
That's what I use. I think they're easy and stuff. So, do you want, there's one there if you want it.
Perfect.
I think do you need the lights on, Mike? Yeah, I need that. No, the one's here. Is that okay? Yeah.
Yeah, it's good. No.
Just straight that out. Yeah. Okay. All right. Thanks. And Lillian. Okay.
Hi, so my name is Siobhan Hammond and I am a medical scientist working in the clinical biochemistry lab at Galway University Hospital. I'm here to talk to you today about what I've done since graduation and how the course has helped me get me there, basically. So just a little bit of background. So I studied here in GMIT between 2010 and 2014. I then did my 6-month clinical placement at Galway University Hospital between 2012 and 2013. And then in 2014, I completed my final year project in the clinical biochemistry lab, where I looked at the analysis of beta-hydroxybutyrate. So then looking at what I've done since graduation. So in March 2015, I initially took up a medical laboratory aide post. Then luckily 1 month later, a medical scientist position became available, which I subsequently took up. And then after 9 months of training and rotation around the laboratory, I became a member of the on-call roster. So then, September of 2015, the same year, I began my 2-year Masters in Biomedical Science through the University of Ulster. So I'm in the second year of it at the moment. It is a part-time Masters online, so that way I'm able to work full-time and still do the on-call while gaining some qualifications as well. So then looking at my current role at the moment as a medical scientist, I kind of break it down into 3 parts, looking at the pre-analysis of samples, the analysis of samples, and then the post-analysis of samples. So looking at the pre-analysis, so basically when you get a form and a sample down, you want to see, do the patient's demographics match the form and the sample? As well as that then, do you have the correct specimen type in front of you? Do you need an EDTA? Do you need a fluorodoxylate? Do you need a serum? Are they correct specimen types for the test you're about to perform? As well as that then, you'll be looking at the time of the sample has arrived down to the lab on time. Certain tests are time specific. Some need to be arrived down to the lab within 15 minutes, an hour, 2 hours, etc., etc. As well as that, in terms of pre-analysis of samples, we have different instrumentation in the labs. So Galway University Hospital in biochemistry is quite busy. So we have lots of instrumentation and analyzers to help us get through all the samples. So operating the analyzers as well is part of some of the pre-analytics. So we have an MPA which centrifuges our samples, decaps the samples, aliquots the samples, labels them, recaps them again, and archives them for us as well. So running these analyzers as well is also part of our pre-analytics. So then looking at the actual analysis of samples, so when you're analysing samples on these analyzers, you need to be running frequent QC to make sure that your different assays are in control, basically. As well as that, part of running the analysis of these samples is looking at your reagents. Do you have enough reagents on board? Do you need to make some more reagents up? Are they stable? Have they expired? And once these checks are in place, you're happy to run your samples on the analyzers. So then looking at the post-analysis of samples, you're talking about validation and authorization. So in terms of validation, are the results you're producing, are they precise, are they accurate? If a result is high or low, it might have had to repeat itself. Are the results reproducible? In terms of authorization then, you'll have a set of results in front of you, and you can compare them to the patient's history. Do they correlate with the patient's history? Are you satisfied that these results belong to that patient? Has the patient deteriorated? Do I need to contact somebody, a doctor, a nurse, or somebody on that team? As well as that is the sample contaminated? Has it been contaminated with EDTA? Has drip arm occurred? And being able to recognise that and respond appropriately. Just having a quick little overview as to maybe why you might decide to do a Master's in Biomedical Science. So once you've all finished your course here in medical science, you're qualified then to become a medical scientist. If that's the career path you decide to choose and you want to further your progression in the medical science career, the next step then is a senior medical scientist, which you do require a master's for. After that then you're looking at chief medical scientist and then laboratory manager. So then in terms of how GMIT has prepared me for my academics and the job I'm doing at the moment. So the degree here that I did prepared me extremely well for the master's I'm doing at the moment. In terms of the topics I've covered, the modules I've covered, there was hardly anything new in the master's I'm doing at the moment that I hadn't touched upon while doing the degree here. Like last year, the subjects I would have done would have been clinical chemistry, DNA technology, things I'd studied here in GMIT. This semester I'm looking at quality management, statistics, once again all topics I have covered here in GMIT. So the degree here does prepare us extremely well for doing a master's. So then looking as well practically how we're prepared here as well. So as we all know, we do many hours of laboratory work here in the lab. And it's great exposure to practice all the techniques that we're going to be using in the laboratory. Doing your pipetting, dilutions. And while these might sound like basic things, you won't be taught these once you go and work in a lab. They're things you're expected to know how to do once you get in there. So use the time you have now to practice all these different techniques. Then looking at presentations and posters and reports, once again, these are skills you might feel, oh, I won't use these once I leave college. That isn't the case. If you work in a laboratory, you'll be doing validations, you'll be doing audits, and you will be expected to compose a report afterwards, to present it to your colleagues, and maybe even make a poster and bring it to LabCon or Biomedica. So, these are practical skills that you will be using. So it's great that GMIT prepares us to, it forces us to do these skills so we can use them outside after the degree. And then lastly, I have clinical placement and final year project. They are in capital letters for a reason. I think we are extraordinarily lucky that we have the clinical placement and final year project integrated into the course. It's fantastic that some of the courses don't have work placement included. So I would have done my placement in Galway University Hospital, my project in biochemistry, and that's where I'm working now. And the same can be said for many of my colleagues who have done their projects and placement in different colleges or different hospitals, and that is where they're working now. So I think it's a great opportunity that we have doing the placement, doing the project, and it does help us, potentially gain a job afterwards too. So then I'm looking at advice to my student self: ask more questions. The lecturers do not bite, so I've survived 4 years here, so definitely ask more questions. Use the services that are available to you as well. I don't think I used the library services properly until I was in 4th year. They do seminars on EndNote and different softwares as well. There's the IT department downstairs. You've got the library, the study rooms upstairs as well. Don't use those. Use them from 1st year. I think had I availed these services from 1st year, not just in 4th year, some of the tasks I would given I might have found easier to do. And then lastly, get the basics right. I know I've said that already, but your pipetting, your dilutions, these are things that you have time to practice them now. Your terminology, accuracy, precision, sensitivity, specificity. Get them right now, because these are words that are being used day to day in the lab, and you don't want to get them mixed up or not know what they mean. So practice them now, get them right now, basically. So thank you, and is there any questions? Yeah.
So you're doing over 2 years.
Yeah.
Do you find it difficult doing the master's over 2 years when you're trying to work then as well, or?
It's tough going, but I think they realise that, I think they've designed it that you should be working while doing it, so that the work you're getting is proportionate to someone who is working full time. So yeah, it is tough going for sure, but it is manageable.
Anybody else have anything that you'd like to ask Siobhan about the lab or? Yeah.
Do you think taking on the lab aid job helped you get the full-time job, or?
Yeah, I do think it was my foot in the door, I suppose, so that when the medical scientist position came up, they thought of me because I was there, basically. I do have colleagues as well who started off as lab aides and then progressed into medical scientists once the positions became available.
Yeah, that is something I suppose we've seen that with a lot of graduates. Lab aides, particularly maybe when you're coming to the end of your 4th year project, if you're in a hospital, coming into the summer they may get funding for a lab aid position.
Yeah.
Or some of the third years guys, when you come to the end of your placement, sometimes the placement hospital might offer you a lab aid position. If again it's resources usually might be the limiting factor there, but it is experience, and I suppose you can't beat that kind of experience. It's a lab aid position, it's not a medical scientist position, but it still is some experience in the hospital. So if that's what you're interested in.
Yeah, I definitely had classmates who would have volunteered even a few hours a week, and once again they were remembered when positions were coming up and things.
Yeah. So when I keep going on about solutions, however much fun the second year is get experience.
Siobhan, can I just ask, just based on that what Mary was saying about the practical work that you do here, we've just done a visit to the hospital where we met you last week with the first years, it's the second year of doing it, and one of the comments from the first years, because they haven't seen, I guess, any of the medical science disciplines yet, they will get a flavour for it in semester 2, and then they'll study them in year 2. Feedback, or comments that we got last year, and I heard again this year, they're always amazed by the amount of automation in biochemistry and the analyzers, and that it's so busy, etc., etc. But I guess, guys, Siobhan here is an example today of a medical scientist that has gone through, and it's really good to hear her talk there about you have to know the basic principles. And I know from a micro perspective as well, you're sick of hearing me saying that things are becoming more automated, but you have to know how to do things manually.
But it's funny, even today I was coming over on the bus, the Merlin bus coming over here, and my colleague was with me, and earlier this week one of our instrumentations did break down. We had to go back to basics, getting the means, getting the standard deviations out, and seeing where assays can control or not. So it was bringing it back to basics again, so it is important. It is true though.
But that's really good, because the secretaries haven't done clinical chemistry yet, and that would be the 2 last years that they taught. Biochemistry was just this room full of analyzers that does all the work.
It's not the case really,
Is there anything else, guys?
Ask a very quick question, Mary, and I promise to stick right after this. Do you use Westgard QC rules by any chance?
I won't be too familiar. I know haematology do for sure.
Oh, that's okay.
That's fine. I should know what biochemistry do now, but I'm not too sure. But haematology, they definitely do.
Okay.
Yeah.
Is that okay, guys? Is there anything else that anybody wants to ask Siobhan? And please, there is no question too simple.
As I said, ask more questions.
Yeah, and if you don't, if there's something, we will have a bit of a discussion at the end. If there's anything that you kind of think afterwards that you want to ask specifically, or if you're too shy, you can ask. Thanks a million, Siobhan. Thank you so much. You should know better that I wouldn't have anything. Actually, yeah, I should have got. See, this is you now coming from private. That's what it is here. Look, you want to take that before I Okay, guys, so I won't. Ger O'Halloran, who is currently in the Galway clinic. So, thanks, Ger.
Hi, I'm Ger. I work in Galway Clinic as a laboratory IT system analyst, and I'm a graduate of medical science. I graduated here in 2013, and I went to America then for a while for the summer, which I'd all definitely recommend. After the final exams, you definitely need to get away for a while. I came back then, and then I had to get a real job, and I got a job in biochemistry in the University of British Columbia working. And I lasted about 4 months there before I lost my mind doing the same stuff. Like Debbie was saying, it's very automated, samples on, samples off, authorise. I couldn't hack it, so I went into Pat, Siobhan's boss, and I said, Pat, I want to further my career, I want to further my education, but don't aim to do a Master's in Medical Science. And we came up with a few ideas, and he said, what would you think about IT? And I started to think about it, and I went back, and I realised some days I spend 90% of my day in the laboratory on a computer, working on a computer system that I know nothing about. So I said, I want to find out something about this. I started looking up courses, and I came across this health informatics course, a master's in the University of Limerick. I'll talk a bit about health informatics in a minute, but it's basically the application of IT to healthcare. So any computer systems and stuff like that, and applying it to healthcare. In the meantime, then I decided to go through some more torture by getting a job in Blackrock Clinic up in Dublin. So I was going from Dublin to Limerick to Galway. And I gave Siobhan my job in the university. And then while I was studying, I started the Masters and I was working in Blackrock. A job came up in the Galway Clinic, a laboratory IT system analyst position, and I didn't have a clue what a laboratory IT system analyst was, but I said it sounds like something I do, so I contacted them and I went down for an interview and I managed to bluff my way in there, and I'm there now still. I graduated from the Masters then a few months later. So health informatics, what is it? It's basically the application of IT to healthcare. So all your computer systems that you see that are worked on by healthcare professionals in the hospital, and or your electronic health records, anything IT or that's done on a computer, it basically covers all that. It even covers cloud computing and other areas like that, but it's nothing like ones and zeros or anything like that. That's not what I do. When you think of IT, I know some people might think of that. That's not what I do. It's more of a system analyst position where I analyse processes and see how I can apply IT or computer system to help those processes be more efficient, accurate, and retain data better. The Masters itself was 1 year long. It consisted of going to college on a Friday and Saturday, and I was working as well. It's considered a full-time course, but you're really only attend part-time. And it includes modules like project management, health electronic records, requirements engineering. It's basically just all IT-related stuff in healthcare. So in order to describe what I do, I kind of have to talk about the Galway Clinic and what we do as a whole as a hospital. The Galway Clinic has been awarded a HIMSS Stage 6 standard. HIMSS is the Hospital Information Management System Society, and it has standards that range from Stage 1 to Stage 7, and we're at Stage 6. And we were the first hospital in Ireland and the UK to achieve that, and we're still the only hospital in Ireland to have that standard. Basically means we're a paper-light hospital, and what I mean by that is we record very little data on paper. It's all recorded on our computer system that we have. Our computer system is called Meditech. It's just the makers, they're a Boston-based company, but it's a hospital-wide information system. It's used in pharmacy, it's used in the lab, it's used by doctors, nurses, radiology, all across the board, and everyone puts information onto the system. And by doing that, we're able to produce an electronic health record. An electronic health record is basically a health record for each patient that's recorded electronically on the computer system. The big difference between those and the university is where you'd have a paper chart in the university and you have all your stuff written down, you can see them into the beds and stuff. In the Galway Clinic, we have computers that we are able to bring into each patient and record the data there, and it's all on the computer. So if anyone ever wants to check up something about a patient, you can access all their information from anywhere in the hospital just by logging onto the computer. You don't have to go ringing down for a chart or anything like that. The data is all captured in one system. That means it's usable and accessible. So by usable, I mean any information that you put into it, you can take it out. You can use that for either just finding out trends or information on a particular patient, what medications they're on, what their lab results, what their x-rays, and like that. And even doctors have this on their phone, so they can look up the patient on their phone, they can put information on that. The data is also usable, as in we can compare standards across national standards, so we can pull information such as the number of MRSAs that were acquired in the hospital, and compare them to other standards across Ireland and Europe. By having all this data on one system in an electronic health record, it helps clinicians make better informed clinical decisions. So you can imagine as a doctor, you want as much information as possible on your patient so you can make the right decision. By having all the information on one system, the doctor can have that all there in front of them and they can look up what they require. And my job really is to ensure that all the data that we produce in the laboratory is accurate, complete, correct, interpretable, and that the doctors are able to interpret it in the correct way, and that they're able to make better informed decisions. So this is just an idea of what our information system is in a diagram. The laboratory system's in yellow there because that's the one I'm in charge of. But you've got pharmacy, you've got registration, you've got doctor, you've got radiology, there's billing because we're a private hospital, and there's nursing as well. So when a doctor or when a patient comes into hospital first, if you think about all the steps that a patient goes through, first of all they're registered, they're asked a few questions: were you ever in hospital before? They're given an identifier, an MR number, it's a medical record number that stays with that patient. And they're asked other questions such as maybe have you ever given birth? Have you any known conditions that you're aware of? Stuff like that. And that's all recorded in the system. So that's available straight away then for a doctor to look up when the doctor meets that patient for the first time. Doctor then can dictate information on that patient and notes through their phone. So they can actually talk into their phone, record notes, and that goes into their record. Any x-rays, CT scans, anything like that, that's also recorded on the system, so they can look it up on each patient. Any nursing notes, any nursing documentation that they record, any nutritional information that's needed to be recorded, all that is recorded in the one system. Just to give you an idea of how important what I do, so we have all this data on the computer system, but it's important that it's accurate and correct, but it's also what we use, how we use that data to help clinicians make better informed decisions. So we give them the information in a way where they can interpret it better. So for example, this is just a snapshot. This is all test information, by the way. It's nothing, you're not looking at any patient in particular results. You all know my date of birth now after this. But, so this is just an example of how the results present to medical scientists, first of all, in the lab. And you can see here, these are, I don't know if you can see there's colours here, but the potassium there, the K is in red there, it's 6.5, it's high. So that's to alert the medical scientist, first of all, that's a critical result that needs to be alerted to the doctor. That also goes across the doctor system, the doctor views it in the same way. So that's an alert straight away. It seems simple just to have colours, but a lot of information systems don't have that. They're in black and white, some of them, they're very old systems. So that pops up and you know straight away then that that's an urgent result. I need to take another look at it and I need to phone a doctor maybe about that result. But just in case you forget to phone it or you haven't been aware of it and you're just clicking through, which can happen in some hospitals, and you're doing so much stuff you don't have time to be taking a close look at all the results, we have a pop-up box. So you can't actually leave that specimen without actually entering in something into this pop-up box that tells you that you have to phone it, records the time, who you phone it to, and if readback was received. So there's no way, and if you don't fill that, you have to fill in something in there or you can't go forward. And also, just in case that doesn't get the message to the doctor, we also send a text message to the doctor. So this is just a text message that's been sent to my phone, and just on the message that just sends the identifier of the patient. It says the potassium is at 7.0 and it gives the reference unit. The doctor then can take out his phone, he can pull up the record on his phone and he can view the patient's details, or if he needs to be anything, any action taken. Another area where we provide information and use the information that we have to make better decisions, get clinically informed decisions, is, for example, a CT scan. So in a CT scan, they often inject contrast material into a patient to give for the x-rays so they can better contrast in it. And when they do that, there's always a risk that if the patient has pre-existing renal problems, that they may go into renal failure. So they have to carry out a creatinine and an EGFR. If they have had a creatinine or EGFR within the last 24 hours, it automatically pops up there in front of them, so they know when they're ordering the CT scan that this is the result, is it safe? They're able to assess it if it's safe to proceed or not. Another idea for a doctor, we have clinical panels. So a clinical panel is basically where if doctor is given so much information in our system, it's very hard for them, like what's relevant, what's not relevant. So in this example, we have a panel, it's a dialysis panel. So if the patient's on dialysis and the doctor, all he wants to know is how he's getting on with his dialysis, is he progressing, how is his bloods looking. All the doctor has to do is click on dialysis and it pops up all the relevant results to the doctor. So they're able to make an assessment straight away and they don't have to go looking through different fields and stuff, looking for laboratory results or having to go through all this information that's presented to them straight away. There's loads of other stuff that I could give you screenshots of and everything. I'd be here all day. for example, if a doctor orders a drug level such as vancomycin, gentamicin, and if they try and give a doctor or give a patient, if a doctor tries to give a patient vancomycin, they're presented with the latest vancomycin level, so they know what dose to give the patient, or if it's safe to give the patient the vancomycin or not. Hemoglobin results appear to the doctor before they order red blood cells. If they're trying to prescribe insulin, the glucose results appear to the doctor, so they know whether to give a high dose, a low dose. Any drug-to-drug interactions when a doctor is prescribing drugs, if there's a chance of it actually interacting with another drug that the patient's already on, that's notified to the doctor. Any antibiotic resistance, so a patient might be on antibiotics, doctor might give a patient antibiotics for an infection, might also take a swab at the same time. It might take 2 or 3 days for that result of that swab to come back, and when it comes back, it might turn out that the patient's actually or the organism is actually resistant to that particular antibiotic that the patient is on. So that's alerted straight away. Once it's resulted by the medical scientist, it's alerted straight away to the doctor. And the other thing we have is all the point-of-care devices, such as your glucometers, your blood gas analyzers, your pregnancy tests that are carried out. They're all interfaced or connected with the computer systems so that all the results populate once they're done. How has medical science helped me to where I am? Well, academically, you all know it's a really tough course. It's hard to get through. Took me 5 years to get through it, so I know I'm not bitter at all, Mary. But when I did go to do the Masters, I actually found it relatively easy compared to it, surprise. But it was actually very good because I was well prepared in doing assignments, handing up reports. So when it came to doing 2 days in college every now and again and handing up a few assignments for a Masters, it actually came very easy to me. Even stuff like referencing and stuff like that, stuff that I learned throughout college. You'll use it if you ever go back to college after this course, if you ever want to do something else. It is very useful to have, and you get a lot of it in medical science, so you do know it inside out once you leave. 3 skills that I just thought of that you have to have as a medical scientist and that you learn throughout the course here. You have to be a quick learner, you have to have good attention to detail, and you have to analyse situations such as results or even processes and workflows. And they're applicable to any job that you actually go into. Every employer is going to look for you to be a quick learner. He doesn't want to be sitting there beside you, holding your hand while you're doing stuff. When I walked into the job in the Galway clinic, I didn't have a clue what I was doing. I bluffed my way into it. I had to learn it all myself, and I just did it. I picked it up quickly, and I was able to mess around with stuff and figure it out myself. Attention to detail, I use it every day now. With troubleshooting, we do deal with a lot of IT issues and troubleshooting stuff, and even when I'm analysing workflows and which details are important, which aren't important, it's very important that it falls into analysis of situations as well. So advice to my students: pass your assignments, hand up your lab reports, and you won't end up doing 5 years, and do your exams. And look, I can't really give any more advice as a student, but when you leave medical science, that's when I suppose your real decisions have to be made. And medical science as a course, when you leave it as a medical scientist, you're not confined to the lab. Don't think of it just sitting in a lab pushing buttons on an analyzer or swabbing out plates, or there's more to it. You can use medical science. I'm working in an office now, and I'm in and out of the lab all the time because of over the lab system, but I'm not sitting down plating up stuff and stuff that was doing my head in when I was working in the biochemistry. So don't think that you have to work in a laboratory setting, because that's not the case. There are other options out there, and don't be afraid to go out there. I did a Masters in Health Informatics. There's loads of Masters out there that you can do once you leave. Don't think that you have to do a Masters in medical science, because there's loads of other Masters that will take you. You have a science degree, so other courses or other Masters will take you. So don't be afraid to try something new. I could be back here in a few years talking about other job that I'm in, because I might decide to do something else. So just don't be afraid to try something new if it's not for you. If it is, fair enough, but it wasn't for me, and I went in a total different path. So it is doable. And that's all I have. Thanks for having me.
back here. It's testament enough to know. So,
Yeah.
Is there anything anybody would like to ask?
Just one down the back there, yeah.
Yeah, do you always have interest in technology before the masters, or was it just you used it?
Yeah, no, I didn't really. No, I wouldn't have thought of. I'm not a computer geek or anything like that. I wouldn't, and I don't do really programming or anything like that. That's not really what I do. I analyse workflows and I see how can I get the computer system to help you with your workflow, so that we record as much data as we possibly can, and make the workflow more efficient. And we have basically the main goal of all this is for the patient. If we have all this information on a computer system, it's always usable by clinicians and to make better decisions for the patient. I didn't know. I picked the masters because I was interested. I wanted to get out of medical science and go into another area. It just so happened it was IT. I was looking for something else, but I'm not. Yeah, you don't deal I'm dealing with computer systems, but I'm not really on a computer all day, so it's not really that techie as such. Yeah.
Anybody else have anything that they'd like to ask Ger?
I don't know if this is for you, Ger, or maybe is this something, this sort of system, is it going to be applied in the public hospital as well? Or
There's a national laboratory information system being introduced across all public hospitals at the minute. So there is going to be jobs being created for laboratory analysts and stuff within healthcare over the next while. The system that they're introducing, they plan to have it connected with all hospitals. So if you want to look up blood results on a patient that when they were in Cork in a hospital last week, you can do that. That's been introduced. It's starting to roll out. It's supposed to be this month in St. James's, but I think I'm not sure if it's going ahead or I haven't heard. We're well advanced in terms of
That's
IT.
just a laboratory system though, isn't
That's
it?
just a laboratory system.
There isn't a central hospital system like what you're
No.
talking about there.
This is an EHR, electronic health record. We're the only, we're the most advanced hospital in Ireland with the system. I think the aim with the HSE is to actually to get to that level, but at the moment they're very far away from it. Yeah.
I suppose we're seeing the difference between when you go on placement or when, for those of you that have been on placement, it's the public versus private, and the resources are the big issue there. Can I just ask you a question though? I mean, you mentioned, did you have to change any of your analyzers? I mean, was everything already interfaced, or was the system that you had just upgraded to feed in, or was there issues there?
For the connections of all the point of care stuff, is it? Yeah, so the system can actually receive messages from all the devices. It's designed that way. That's what it's there for. It's the hospital information system. There wasn't many issues. What you have to do is you have to contact the person providing the device and contact your computer system, which is Meditech. And between that, then we come up with a messaging system, and then the messages just flow in. It's through an interface.
How often does the system go down?
The computer system as a whole doesn't. We have scheduled downtime, but it's never gone down while we've been there. There's servers. The servers are actually on site. They're not external, so we have control over that as well. So it's very sound structurally. So we haven't actually ever had any unscheduled downtime. And when there is scheduled downtime, or when there's unscheduled downtime, there's always downtime procedures in place in the hospital. So a computer system should not stop you. If it goes down, it should not stop you from carrying out your job. And it's only there to assist you. So it's there to assist you, retain information, but it should never stop someone from carrying out their job. So there should always be a way of doing your job without actually using the computer system. Yeah.
Sure, I have, it's a comment rather than a question.
Yeah.
I think it's interesting to hear you've said once or twice, and just for everyone, again on this, I've heard you say, I wanted to get out of medical science, I wanted to leave medical science. Listening to you up there this evening, the way you can talk about results and tests, you're still in medical science now, but you leave.
Yeah, well I suppose when I say that...
Well, no, it's fantastic,
Yeah.
you're in No, it's flowing, different test results, analysis. You're gone to IT, but someone with an IT degree would not be as good at the job you're doing. You know the background to all of the tests and all.
Yeah, and that's true, and it helps me big time in my job. I wouldn't have the position now that I'm in, because I had no previous experience in IT or anything like that. I was just starting a master's. And when I went for the job, and I just chanced my arm really. I said, sure, I'll have a go and see if I get it. But it was my lab experience. I knew all the tests, I knew the processes, and even it helps me big time when I'm talking to someone in the lab trying to come up with workflow. They're able to tell me stuff, and I understand what they're talking about. They're not trying to speak about those.
That's an important point.
It's huge.
It's the routine
And I suppose when I say I was trying to get out of medical science, I wasn't trying to get out of medical science as such. I was trying to just get out of the routine, the routine lab,
And I think that was something we would always say. I would say to students that I would meet at open days, I might have met some of you, is that it's a fantastic foundation degree. It has the added bonus of allowing you to be a medical scientist if that's what you want, but you have fantastic foundation to go on into other roles as well. Did you have a question?
Yeah, just I found it too much, results down to doctors and their phones and things.
Yeah.
And we know like in university sometimes there's any issues in terms of like data protection and things, results down to their own doctors and
No, the way it gets through the data protection laws and stuff like that is when the text message, we send the text message, we send the identifier, which is a hospital identifier for that patient. So we send the hospital identifier and we send the test and the result. There is no way of identifying what patient, or there's no way of a randomer picking up that phone and seeing that text message and being able to say, oh, that's Gerard Hollern down the road, he's a potassium here. There's no way of saying that. You have to have access to the computer system and to actually be able to identify the patient. So that's why it's allowed. There's no way of identifying who it is unless you have access. And in order to have access to the computer system, you have to have a log on and a password, and it's security that way.
Do the doctors actually like the system?
The
fact that they're getting text messages while they're on the golf course?
It's an opt-in, opt-out. We've actually had them get text messages out on holidays in Spain and everything like that. They do ask to get turned off at times. It's an opt-in, opt-out option, so a lot of the doctors do opt into it because they actually like getting the text messages. They're real time and they know when something's happened to the patient. There is some doctors that don't. So the likes of the oncology doctors that will be dealing with them, they don't like it, mainly because every result, if you're an oncology patient, is going to be out of range, so you're going to be getting loads of text messages. It's more the other general clinicians that actually use it and they do like it, yeah.
Would you on call there as well? Like if the system did go down, would you be on call for that?
There's an on-call system where so I'm over the lab system, but then there's someone over the nursing system, doctor system. So there's a team. I think the team's up as 17 at the minute because we're putting in a new system that's going to allow us to do more things other than just what I've showed you there today. So we do an on-call system where it's a week on, but it's standby on call. You have a phone, week on, week off. So there's an on-call system. But if a system did go down, everyone would be in because it would be a big thing. Yeah.
Okay, is that it? So are we happy to move on? So
Sorry, I took it off.
all right. Brilliant. Sharon, Stephanie's going to come up. I'll let you go. You go next. Is that okay? Not at all. Come on, I hate you. Okay, so we'll move on to another slightly different position as well. So I'll leave you with Stephanie. Thanks.
Thanks, Mary. Hi, everyone. My name is Stephanie Moyles, and this evening I'm going to take another path like Jura, but mine is even more veered off the routine hospital path. So I'm actually going to talk to you about the pharmaceutical and medical device industry and my career through both of those actually at this stage. So as I said, my name is Stephanie Moyles, and I actually graduated from medical science here in GMIT in 2010. And after that, well actually before this, like most people at that stage, I hit the road, I flew off to Australia. I had been travelling every summer anyways, instead of staying in the hospital and doing my lab aid work for free. So I couldn't wait to get going. And off I went thinking I'd just walk straight into a hospital there and be a medical scientist and go to the beach every day and it'd be great. But I wasn't long there when I realised that really without placement over and above, you know, your without experience over and above your placement that you would have done as part of the course, especially in a different country, you're not really going to get far. The other thing that I did meet a lot of were questions about whether my qualification was transferable in Australia. So definitely, if you are thinking of taking your medical science degree elsewhere, I just make sure that you have all of that kind of your T's crossed and your I's dotted before you head off, so you don't meet any unnecessary questions. So I came back from Australia, and there was HSE embargoes and there was slim pickings. There wasn't much in the private hospital, and there definitely wasn't anything in the public hospital unless you're willing to go in and work for free, and that's not what I was willing to do. So I figured I better widen my search, and I thought, okay, well, what do I widen it to? What else can I do? Unfortunately, we didn't have an evening like this when I was here, and you guys really are lucky to be able to get a wide variety of what options are available to you. The only thing that I did have in my head was Mary McGrath telling us that when she was qualified and there was a recession, that, you know, they went off and went into industry and it was all fine because eventually the hospital lads can call in and there was jobs everywhere. And so I thought, okay, yeah, I'll go into this industry, crack for a while and see how I get on. So then I thought, okay, all right, I'll apply for a job, and again, didn't know what I was applying for, and in fact I actually applied for one job I was about 20 years experience and a PhD short for, but that's just, a qualified person doesn't just mean a person who's qualified, but that's for another day. So I went around with my CV and I got an interview and actually a job pretty quick even, in Baxter in Castlebar in the microbiology lab, and I was there for about a year and a half. And I moved then to Milan, which was Bionish out in Invern, and I was a microbiologist there as well. And the industry lab, be it pharmaceutical or medical device, isn't too dissimilar actually from a medical microbiology lab. I suppose one of the big differences would be your sample type. So you're not looking at feces or urine or bodily fluids. You're going to look at water or products or your environment. So so in your pharmaceutical lab especially, you often may have products that are manufactured aseptically. So this means that they're not going to be thermally sterilised. And in cases like this, your environment is particularly important. And by environment, I mean not only the manufacturing environment, but also the personnel that are manufacturing the product. So in this case, if you have an issue with, say, bioburden or surface bioburden or air burden or particulate in your clean room or on one of your personnel that are touching the product, and this gets into a product that's not thermally sterilised, it's a critical significance really and impact. So this needs to be continuously monitored and results assessed and any impact acted upon. So and then from the medical device perspective with microbiology, some of the sterilisation techniques that we use, for example irradiation, there's actually a direct correlation between the bioburden that's on your product and the particular E being the strength of your electrons and strength of your dose that you're going to give the product. So in cases like that, if there's a change in your bioburden, you can actually no longer have a process that's sterilising. So that's very important to monitor and to keep control of. So I was in the lab in total, I suppose, year and a half, it's about 3.5 years, nearly 4 years, and I was itching to get going again. And so I looked at my options, and I found myself moving not only from pharmaceutical to medical device, but also from a lab-based job to a desk job. And there was a couple of different challenges within that. Obviously, pharmaceutical to medical device is a whole different ballgame. You kind of think of it all as industry, but it's not. They're very different standards, very different auditing bodies, notified bodies. So there's a lot to contend with. But actually what I found to be quite a challenge was to move from a lab-based role to a desk job, and especially because I was moving into validations. And the challenge I found here was because in a lab, usually you've quite daily tasks. So you come in the morning and you know, okay, I need to get this test on and get this test off and get the results through. Whereas when you're in validations, you could have a project that's ongoing for months or even years or even is never ending. So to manage your time on that, it's a funny one to get your head around. But it's definitely something that I enjoy, and I'd never go back. Validations is a great area to be in, and you're constantly thinking, and there's a lot to go with it. And there's so many different areas. So it's definitely an area that I would recommend to anyone that would be interested. So first of all, I was in Boston Scientific. I was there for just short of a year, and I've since moved to Medtronic, and I'll be there 2 years in February. So Medtronic, for anyone who doesn't know, we have one site here in Galway. It's the biggest medical device company in the world, especially since acquiring Covidien. There's also a site in Galway, but that's now known as Medtronic. One of the biggest businesses in Medtronic, and what the Galway site is involved in, is cardiovascular disease. And as part of this, we manufacture drug-eluting stents, balloons, stent grafts, inografts, so a lot of different devices. And I won't go through them all now because we'll be here all night. But if you have any questions, if you want to come up to me afterwards, or obviously you can check out the website. I'm not selling any jobs here or anything, but so my current role is I'm a quality engineer in Medtronic, and I specialise in sterilisation, validation and biocompatibility. So essentially my job is to make sure that the product is safe for the patient that it's being either used on or being put into. And what I'm looking at as regards that safety is it sterile? In other words, are you going to put that product into a patient, and especially, most likely that patient is going to be immunocompromised. So you don't want to put in a product that's full of bacteria or, other that's going to go in and cause a pyrogenic reaction or infection. We also want to verify that the product is biocompatible. So in other words, you don't want to put in a stent that's going to be there long term in a patient, but it's actually going to cause them cancer. So you don't want to solve one problem and cause another one while you're at it. So it's very important that we look at these two areas. So typical tasks in these two areas would involve, first of all, validation. So we need to validate a process by which to sterilise the product. We don't validate biocompatibility. So the materials of a product are either biocompatible or they're not. So biocompatibility is more of a yes, it is, and then if there's changes, we assess them. Whereas sterilisation it can be quite an ongoing process, because you can validate a product, but you're validating that product as it is, how it's currently manufactured, and how that sterilisation process currently is. So if there's any changes along the way, you can impact on that, and it may seem minor, but the end result can actually be a major impact on the patient receiving the product. So we do an initial validation, and that validates what the sterilisation process is on a routine basis for a commercial product. And then we reassess and we revalidate that particular process for that particular product on an annual basis, and that's ongoing. And then we also assess if there's any, as I said, any changes can have a major impact. So we assess any changes to the product, and this may be, something significant, like are they changing the metal in the stent, or even as simple as the change in the ink that's in the carton on the outside of the pouch of the primary packaging. And then a huge part now, and it's becoming a bigger and bigger area for us, is audit support. So medical device companies are subject to audits from regulatory agencies and also notified bodies. So we're regularly audited by several different bodies, and of course microbiology and indeed sterilisation is such a huge can have such a huge impact on the product and the safety of that product for the patient, that it's always asked. There's always a question from every auditor on either microbiology or sterilisation or both. So definitely we're heavily involved in audit support, and that's never going to change. So some of the technologies that are employed for sterilisation in industry are ethylene oxide, which is a gas. There's also irradiation, for example, gamma, E-beam, and then we also use steam or dry heat. To choose your technology, that's definitely the role of the SME, but it's not singularly the role. So you're usually at the beginning, at the forefront of a new product development, you'd have some sort of discussion and correlation between the R&D group and the sterilisation group and biocompatibility group to make sure that you're choosing the correct technology. And often it's not a cut and dry, oh yeah, this one ticks all the boxes. It can be, how many boxes can you tick that this is the best technology. So for example, if you had a product that's made composed of materials that are quite likely to degrade or are highly degradable, you definitely would not go for an irradiation technique. And you look then, and similarly, if you had a product that had a high bioburden count, you might prefer to go for a more robust technology and not going overkill like ethylene oxide. But again, that's kind of more technical there, so we'll leave that at that. So at the moment, my kind of current role is I'm validating 2 new products into Galway. It's not a usual occurrence for someone to be validating 2 products at one time, but I decided to do this, and the reason for this is because we are continuously working to improve processes. Everywhere, no matter where you work, be it public or private, you have resource constraints, you have cost constraints. You're always trying to improve processes, have cost savings. So what I've done is I strategized that I took 2 new products coming in, knew they were coming in within a year of each other, strategized a validation strategy where I can not only validate the 2 products together, but I will also then be able to allow that product team, that business unit, to have the capacity and flexibility to sterilise those products on a commercial routine basis, either together or singly, and have an option of 3 different sterilisation chambers. So that mightn't sound, anything too special, but for us it's a huge deal, and it means instead of creating more and more cycles that we have to, or processes that we have to maintain and revalidate and constantly on an ongoing basis control, we've just reduced it, by half. So definitely, if it's successful, and hopefully it will be, because I want to keep my job, it's something that people will be doing, more and more of. So it's kind of a trial and error at the minute, but it's a good learning opportunity, as I keep being told. So co-sterilising. So if it might sound like for example, ethylene oxide is processed in a chamber, because ethylene oxide is highly flammable and cannot come in contact with air at all, or else we'll have a huge explosion. So the chambers that we use are in Tullamore, and so what's routinely done is we will put 1 product into the chamber, and that's sterilised on its own. But with this project, we'll actually be able to, the teams and the planners for each of Aptus and atherectomy products will be able to decide, okay, I don't have 12 pallets of product this week for my sterilisation cycle, but you have 6 and I have 6, so let's go together and we'll get a full chamber. So you are, using your full capacity of your chamber, and you're getting the highest benefit from a cost perspective of that cycle. So this, I thought, you think you relay this information to team, but unfortunately sometimes it's hard to push what's the norm, and let people know that just because this isn't the way it's always been done doesn't mean it's not the best way, or that it's not equally as good, if not better, than what's always been done. So so this took a little bit of agreement from the teams, but that again is just another challenging part of your job, and something that it's a good part to have. So what we've been able to do is get a cross-function assessments from design assurance, from design verification, to ensure that there will be no impact to functionality, and also biocompatibility is a concern in this kind of a project, to make sure that there will be no impact as a result of co-sterilising. So these are 2 products that I'm working on, and I just have a short little clip just to give you an idea of some medical device products. So just click into this. So this is Aptus, Helifix, and so as I said, a lot of the, I think I can make this bigger, so a lot of the things that we make in Medtronic and Galway are stents or endografts, And what this does is it actually, I'm going to rewind that because it's actually quite short. What this does is, if we've put an endograft into a patient, unfortunately what can happen, it doesn't happen all the time, but what can happen is we can see migration of that graft, and it's not like it's going from your head to your toe, but even a slight slippage can encourage leaks and cause leaks. So what we can do, as you can see here, I'm just going to bring that back to the start, is we can actually use these anchors. And these anchors can be used either when the endograft is first put into the body, or if the clinician is concerned that there may be migration of an endograft, they can go back in with these anchors. And what you're seeing there actually is the line here is, we'll say, the graft, and this is the vessel wall. So they go in, they apply the endo anchor to the end of the guide, and they come in and literally screw it to the inside of your vessel, so it's not going anywhere after that. So that is aptus. Oh no, I'm going to the wrong one there. And then this is atherectomy. So this is completely different, but nonetheless, I'm going to mute that. So what atherectomy is? So atherectomy is a directional atherectomy device, and what you see here is patient's an artery in a patient's leg that is full of plaque. And I suppose in the past you may have had to go in there maybe with a stent or a balloon or something to get that plaque moving. But what we can actually now do is go in with the atherectomy device. You can see here there's like I think it's a bit like a cheese grater. I don't want to put anyone off there doing it now, I'm sorry. But we're going in here collecting some plaque off the artery wall. It's actually got a collection vessel on the back end of it to collect, to retreat out of the vessel, discard the plaque, and then the clinician can decide to go back in if needs be and collect some more. The result being that the patient, it's minimally, I don't know what I call it, minimally invasive, but the patient isn't left with an implant and is now free to look after themselves healthily. And you can see here on the two pictures, so here's before, you can see how narrow the arteries are showing up, and then here how much room they have for blood flow. So it's just a little example of some of the products that are manufactured, and you can imagine how important these are to the patients that are receiving them. And that patient doesn't want to receive such a great product and they get an infection or cancer or a mutation afterwards. You know, it sounds simple, but it's true. So how did GMIT and medical science prepare me? So this is actually something that from time to time I kind of wonder, why did I even need a medical science degree? Because I never really used it. What I mean by that is I never really needed that eligibility for membership of the academy that, we do speak quite a lot about. But when I actually think down a bit and I think, I could have just done this with a science degree or an engineering degree. But when it really comes down to it, I have no doubt in my mind that my ethics are as strong as they are, and that the level of the standard is so high that I expect from my team members and from myself, and that is in no small part due to studying here in GMIT, to the lecturers and to the course in itself. And because really the whole time it was reiterated when I was both here and also when I was on my placement, was that there's a patient at the other end of that blood sample, or the other, whatever sample it may be, and that patient's important. And that sense of responsibility that I gained here is something I've brought with me through every job that I've had, be it pharmaceutical or medical device, and it's something that I hope I never lose. And thankfully in Medtronic it's mirrored by our one of our mottos is, you touch the product, you touch the patient. So no matter if you're a product builder on the line, or if you're in validations, or if you're senior quality of director, you are touching that product in some way, and so you are actually touching that patient. So what you do every day is very important. And also, it's just, even whether you use it or not, it's nice to have a background in medical device medical science, and to know a bit about disease, whether it's for your career or in general in life. It's nice to have. So if I had one piece of advice, if I was able to go back and talk to myself Well, I tell myself a lot of things, but definitely one thing that I would like to say to myself was to stop worrying about what everyone else was doing and just focus on myself. And I know, we've heard that time since we've been tiny little kids. Your mom would say it, or your granny, or someone, but it's really true. And it's something that it's only now when I look back that I realise how I didn't need to be worrying about. I used to get so down when someone else would get like really, really great marks in written exams. Like Ger, I decided to take 5 years to do my degree instead of 4. And I get so down and worry, like, what am I going to do? And then when I, went into industry, I thought, oh God, am I such a failure? Like, I haven't used my degree. And you get so worried. And I never once realised that actually written exam, they're all finished once you leave college, unless you obviously go on to do further study. And what's been one of my greatest strengths and the reason for my success has actually been my character, and the fact that I'm such a hard worker, that how I treat people, how I come to team meetings prepared, how you use initiative, how you want to improve, how you want to challenge yourself and always keep growing. If you're in the same role, or if you move into a new role, or whatever position you're in, that you're always pushing yourself, and that you're never settling, and that you are always willing to help and be a nice person. And it sounds so simple, but when you're in a day-to-day job and you're working for 40, 50, maybe on a bad week, even 60 hours a week, it makes a big difference if someone is actually a nice person and you're able to get on with them. So, I think that was Dr. Seuss. I don't know that anyone can say it better than him, but the truth is that only you are you, and never forget that. So when, if it's now or if it's in a few years' time when you're out in the real world, just remember that if you're in an interview or you're working, just be yourself. And that, if you're a good person and you're trying hard all the time, that'll get you a lot further than you realise. Okay.
We're all motivated now, actually.
Go and take some more. No.
Has anybody anything that you'd like to ask Stephanie, or any comments, or completely different utilisation of the knowledge that you have? And I do think the foundation that you have does stand to you more than Ger as well.
Definitely.
Between the pharmaceutical and the medical devices.
Yeah.
For those that have just finished third year, a lot of QM words being even they just had their QM assessments the third year.
Yeah.
So validation. So that's a module probably that those sometimes students go, why do we have to do this? I mean, it's extremely important in the clinical setting as well, but certainly in the medical device and the pharmaceutical, there's no escaping it.
Exactly.
That language is used, and again, like Siobhan said, those terms are expected that you're
familiar with them.
Nobody's going to sit you down and tell you.
Yeah, I actually remember my first interview in Baxter. One of the questions they asked me was, "What's an SOP?" And I was like, it came out without me even thinking about it, and it was from just hearing these things over and over and over and knowing the basics, knowing those things.
So we're back.
Is there a lot of repetition in the validation procedure?
Not a bit. No. One product is different, and a product can be one way one day and another way, or process, I should say. So that's actually one element that I love, because it's constantly changing, and it's you kind of gain some ground of confidence and think, yes, you know what I'm doing, and then it all changes. And it's always evolving as well, in line with standards, and by that I mean international standards. And what we realise is maybe better practice or goal practice. As I said, Medtronic is the biggest medical device company in the world now, so we see a lot from the FDA that we are audited a lot harsher. Obviously I was in Boston Scientific before this, and I can vouch for that Medtronic are definitely audited with a heavy hand, and the FDA have admitted as well, or not admitted, but highlighted that because Medtronic is so big, they expect us to be gold standard, and because of that, the standard that they're going to audit at is that much higher. So it's a tough environment, but it's one that definitely like to turn in our favour and say, well, yeah, you can audit us as tough as you want and we'll still do great. You know, so it's just a quality standard for us. Yeah.
Is that okay, Phil?
Yeah.
Can I ask you, is this your lab work?
And not in the slightest. So, yeah. So, yeah, no, I don't at all. The only thing I would say though is that while I have no hands-on in the lab anymore. In my protocols that I write, I'm still developing lab methods. So at the minute, I'm trying to develop a strategy for the apparatus that we saw, so the endo anchors. So the applier that applies them actually has a battery contained in it. So at the minute, I'm trying to develop how I'm going to test that battery in media for 14 days without having a false positive. So I definitely still have my hand in that area of it, which is nice. But I'm just not getting the hand dirty, so it's lovely.
Thanks, Stephanie. Million, do you want some water there?
Yeah.
Sorry,
I thought it was a third one.
I know, that's okay. I think Stephanie's you were speaking and stuff, so that's fine. Maybe we'll just give people a couple of minutes just to talk, not to chat. Something to do there.
Yeah.
That's great. Okay, guys. We'll keep going, guys. So we have just 2 more speakers, and they're all very, very different, as you can see. So again, it's just giving you a slight taste of what's available for you. And I suppose, like Jerry was saying, not everything is for everybody. So when you know your strengths, you can work to that, but you can't beat the foundation that you're going to be leaving, springboarding from here. So I'll hand you over to Sharon. Okay, thanks, Sharon.
Thanks, Mary. Hello, everybody. My name is Sharon Duffy, and I'm just going to talk you through what I've been up to since I graduated. So I graduated from GMIT in November 2014 in medical science, and as part of the medical science degree, as you all know, there's the research project in 4th year. So I was fortunate enough to be able to do my project here in Galway, or in GMIT, in the Galway Medical Technology Centre, or GMEdTech for short, and it's actually located here at GMIT. I didn't even know it existed. So just briefly to explain what the project was about, we were involved in making blood clots from sheep or from bovine blood, and then we developed a histological protocol to analyse them blood clots. But I found that the research project was one of the most valuable aspects of 4th year, because it really developed my research skills, and it also allows you to make that link with industry or a hospital. And also the research project was extremely important in where I am now in my career, and I'll go through that later. As part of the project, I was lucky to be able to present it at the LabCon conference that the Medical Science Academy organises, and then I went on and I represented Ireland in Croatia at the EPBS conference as well. So after college then, I was lucky then to start straight away working in a hospital laboratory in Mullingar, so in the Midlands Regional Hospital, Mullingar. So I worked in blood transfusion there for 9 months. And I just want to highlight that I worked in blood transfusion, but as part of my 4th year I studied biochemistry and microbiology. So I know some people might be worried that, if I studied hematology or cell pat, does that mean I can only work in hematology and cell pat? No, it doesn't. It doesn't limit you at all in what laboratory you can go working in. And also that was my placement hospital as well. So kind of just keep that in mind, that a lot of placement hospitals do tend to take their students back again. And I did work there as a lab aide during 3rd and 4th year as well. So just to go on now to what I'm doing at the moment. While I was doing my research project here in Galway, a medical device company called Neuravi, they're located here in Galway, they approached my supervisor at the time, and basically they just asked my supervisor, did he know of a suitable candidate to undertake a PhD with the company. So he suggested that I apply to the Irish Research Council for funding for a PhD, and I got the funding. So the PhD, I suppose it was all good timing. If I didn't do my project here in Galway, I probably wouldn't be doing the PhD. So, and it is a continuation as well of my final year project. It's an Irish research employment-based PhD, so it's an employment-based in that I'm working with Nuravi, and it's co-funded by the Irish Research Council and Nuravi. And it's a 3-year PhD, so I'm halfway through it now, and I've another year and a half to go, hopefully. It's, as I said, it's co-funded by Nuravi. Just to give a little bit of a background on what they do, they're a medical device company that make thrombectomy devices, and unfortunately don't have any fancy videos to show how it works. But basically a thrombectomy device is a device that's inserted into a stroke patient, into the brain of a patient when they have a stroke, and it's used to remove the clot. So it's a very thin wire that basically goes up through an artery in your leg, and the doctor guides it up through the artery into your brain, and basically they catch the clot and bring it back out. So that's just it in simple terms. I just want to go through what my role as a PhD student is in the company, because I know again it's kind of rare for someone to go into industry or to be involved with industry, but as part of the PhD we developed a range of blood clots that have a range of compositions, and they're intended to represent those that are retrieved from stroke patients. So basically that involved me having to go to the literature, look at any work that has been done on clots removed from stroke patients, and then trying to replicate them clots ourselves. So you're probably wondering, well, what's the point in having all these lovely clots? Like, what's the point in having them? So basically the company is using these range of blood clots to test their devices, and they're developing new devices, trying to improve their device. So in order to test their device, they need clots. The clots are also used for hands-on training with the neurointerventional surgeons who perform the thrombectomy procedure. So in order for them to basically get trained up, they're not just going to go straight in and do the procedure on a patient without being trained up. And then they're also being used in a number of stroke centres throughout the world. A number of different centres are doing imaging studies to see, can you predict the composition of the clot from a CT or an MRI scan prior to performing the procedure. So that's like really useful information if you can tell before performing the procedure if the clot, for example, is a friable clot full of red cells. Well, then they know then that clot is likely to break up and may cause another reocclusion later on, maybe within a couple of days. Or they might know maybe it's a fibrin-rich clot, which will more than likely be extremely difficult to remove. So they may have to choose a particular device, particular competitor's device over another device. And we are also hoping to, I know some people think this is a bit disgusting, but we're hoping to keep the clots that are removed from stroke patients. So once it's removed, we're hoping to basically keep them and perform some studies and testing on them. So Beaumont Hospital at the moment is the only hospital in Ireland, or in the Republic of Ireland, that performs the procedure. So we have a number of collaborations in place with them to hopefully collect the clot, and well, from hundreds of patients, and collect data regarding the case. So we'll be looking to collect data such as if the patient had any underlying disease such as atrial fibrillation, or if they were smoking or something that may have contributed to causing the stroke, the number of attempts made to remove the clot, what other information, the number of pieces that the clot is in, like is it intact as one whole clot, or is it has it fragmented into 5 or 6 different pieces. So then we hopefully then will do, after maybe a year, once all the clots are collected, we'll do studies and hopefully be able to do a correlation study. And I'll emphasise, if anyone here is interested in going on to do research, you'd be very used to doing ethics proposals. They're a bit of a pain, but you have to do them. Yeah, so just to go through how doing the PhD in correlation with an industry is, what advantages basically there has been. So it has given me the opportunity to present both nationally and then internationally in hopefully in a couple of months or in the year to come. But it has also given me that industrial experience. So I've gained my hospital experience through placement and working in Mullingar. But I never knew what it was like to work in industry. Like, I was always focused like on going to hospital, that's it. But it's nice to actually see what it's like in industry, to see how their processes work and all that. So I do spend quite a lot of my time there. I spend about 70% of my time in the company and maybe the remainder of the time here in GMIT. So I know a lot of people are mad to get out of college after fourth year. I was kind of like that as well, but you know, I'm still continuing my education, but not in college the whole time. Another thing I found doing a PhD in collaboration with a company was that the amount of collaborations that has opened up. Like, we're currently collaboration with Beaumont Hospital, as I mentioned, but also the Mayo Clinic in Minnesota and the UCLA Stroke Centre in Los Angeles. And to be honest, if I was probably doing the PhD on my own, I probably wouldn't have made them collaborations. Like, the company has so many links. Like, I definitely wouldn't have been collaborating with all three of them. And finally, it has given me a chance to publish the work as well. So I suppose that's another kind of good thing to have on the CV or whatever. Now, just to give you some advice, I hope it's good advice. Yeah, the first thing I'd say, I don't know if anyone here is actually a member of the academy, the ACSLM. I would encourage you to all just join up. It is free to students, so you've nothing to lose. Literally just 2 minutes, fill out a form, that's it. And I would encourage you as well to take part in the CPD cycle, the Continuous Professional Development. So basically, it's another thing to put down in your CV. It shows that you're interested in the profession or whatever. So it might involve maybe, well, you get points for just becoming a member anyway, and you get points for graduating. So you have loads of points there already, but you get other points for like just doing a short little quiz, reading a journal article and doing a quiz at the end of that. Just little tasks like that might only take 5-10 minutes. And it's another good thing to put down your CV. If you're going to look for a job in a hospital, you can say you're taking part in the CPD cycle. And also, I don't know if you're told or not, but I think in the next couple of months it's going to be mandatory for all medical scientists, well, people working in a hospital, practicing medical scientists, to have core registration. And one thing that's mandatory for core registration is that you're actually taking part in the CPD cycle. So if you start now, give yourself a little bit of a head start. Yeah, and I would also just advise you maybe to attend any conferences or talks that are out there, to just stay up to date with what's going on in the profession, basically. The academy, again, are great. They hold loads of meetings, like they even hold meetings relevant to students, like meetings telling you what options you have out there as a medical scientist, and they hold meetings on how to actually use the CPD cycle, because a lot of people go onto the website and they haven't a clue how to use it, so they just say, to hell with this, I'm not going to bother with it. Yeah, and then there's conferences like LabCon and Biomedica. They're two great conferences to go to. You meet a lot of the industries do go to them conferences, like Abbott's, Beckman Coulter, AccuScience, people like them. And if you're interested in getting into the industrial side of things, Biomedica would be a very good conference to go to, and maybe bring your CV along and give it to them. What else? Yeah, another thing just is probably isn't going to help the fourth years, but for the the first, second, third years, when you do go on placement, I would advise you just to be as willing and as helpful and enthusiastic as possible, because as I said, a lot of people do get their first employment in their placement hospital, and the field of medical science, it's so small in that everyone knows everyone. Like, all the medical scientists know each other. Like me myself, I probably know someone in the majority of the hospitals throughout Ireland, and like a lab manager probably knows the lab manager in every hospital. And if you're going for a job, all they have to do is just lift up the phone and ask what were they like on placement. Oh yes, and this probably does refer to the fourth years. I know fourth year is very stressful and all that, and you're thinking about passing the year and studying and all that. But I would advise you maybe towards the end of fourth year to just start looking at what jobs are out there, and maybe start applying for a few jobs come the end of fourth year. Because when you finish that in May, you don't want to be sitting there with no job. But then people are often stuck, well, where do I go to look for these jobs? The Academy website actually do display a lot of the jobs, so that's another reason why to join. And you have to be a member in order to log in and view what jobs are available. And also the HSE website, they advertise jobs as well. And there's agencies. I think a lot of hospitals might be trying to get rid of agency work. I don't know if there's many agency staff now, but the two main agencies I think are the CPL agency and TTM, depending on what hospital. But I don't know how relevant that is now. Finally then, don't be afraid to continue your education. I know a lot of people just want to get straight out of college into a hospital and that's it. That was a bit like me, and things just fell into place. I didn't think I'd be here, like, to be honest. So you have loads of options. You can go and do a master's. As we said earlier, there are loads of masters out there, but if you're thinking of going into a hospital and looking for maybe a promotional position later on down the line, in years to come, there are specific masters that the Academy do accredit in order to get fellowship. But then there's loads, like, people just think, right, Coleraine or DIT and do the medical science masters there. There are other options as well, so don't limit yourself. And then if you want to go for your sins and go and do a PhD, there are a number of funding bodies out there. I know people mightn't be aware, well, if I want to do a PhD, how do I go about it? There are loads of funding bodies. Mine is the Irish Research Council, but there's also the European Research Council. Science Foundation Ireland is a good one as well. They have a lot of PhD positions in NUI. I know I shouldn't be promoting NUI, as opposed to GMIT, but the Health Research Board as well is another one. Enterprise Ireland, and also Teagasc. They're not just for farmers. They do have some, I think, a lot of microbiology based PhDs and that. I think you just keep an eye on them sort of websites or whatever. So I hope that's advice does help you somewhere along the line. So that's everything. If anyone's any information, I've left my email address there, but you don't have to email me if you don't want to. But if you have any questions, I'll answer them now.
Thanks very much, Sharon. I think Sharon is definitely an example of where things, when opportunities come, that you take. Definitely what was presented to you, but your role just...
Yeah, because originally I wasn't meant to do my research project here, and it's just the way things happened that I ended up doing it here.
Sorry, is there anything?
I'm just wondering, how long does the clots last for when you get to
Sorry?
the hospital? How long do the clots last for? Do you have a certain period of time?
Well, we have it arranged. Well, we've submitted ethics to BOMAT, and I got an email during the week saying more or less it is accepted.
Oh.
But the way we're doing it is that as soon as it's taken out of the patient, there's going to be a nurse there that will basically put the clot into formalin for us, and we'll fill out basically I've done up a sheet with what information we want. They'll fill up the sheet, pop the clot into formalin, we'll ring myself or someone, and I'll organise by Omnis to transport it straight down to myself in Gola. So once it's in formalin, like, it should be okay. Yeah.
Is there anything anybody else would like to ask Sharon specifically, or no?
When the PhD finishes, will you still be with the company then, or?
I don't know. The company mightn't be there. I don't know. Yeah, like, yeah.
Bigger. Nobody will.
Yeah. To be honest, I don't know what I want. I've seen both sides of things, and at this stage, I don't know what I want. But I suppose I'm keeping my options open. Like.
Okay, so guys, we're okay, Rich. I don't know any fancy flickers. Apologies. Sorry about that. Yeah, here, there, yeah. Okay, guys, so we have.
We'll fire away or the tea will be gone cold.
Yeah, we'll fire away or our tea will be gone. Yeah, exactly. Yeah. So we'll go off with Rich. Thanks, Rich.
And thanks, Mary, for saving the youngest till last. So I've obviously got the shortest career of all the speakers, so hopefully I have something for everyone. But this is going to be a bit different to everyone else's because I want you to ask me questions. There's not much point to me giving you stuff that I've done over 30 years and assume that that's what you want to hear. I want you to ask me questions as I'm going along, okay? So essentially, I'm going to give you a snapshot of 30 years working in different aspects of what can happen after the course you're in or going through. So this is what happened from the 80s to 2001. So I started off, admittedly, it's a confession. I didn't know it was only for graduates at GMIT when I was invited. But we'd say nothing, because I've actually living down here for 25 years anyway, so it shouldn't be a problem. I'd sneak out anyway. But I went to a parallel universe, essentially, where we did the same kind of course setup in Dublin as was done here in the West. So I started in DIT Kevin Street, the equivalent at GMIT. Now, initially, it used to be a cert course, 3-year cert course, 2 years in college, and then 1 year actually on the job, you know, in a training environment similar to your 4th year. And my training was in James's, so I got a broad range of lots of different techniques. A very large hospital, similar to GUH, and it set me up basically for anything I wanted to do afterwards. Then after that, I went to Piedmont Hospital. It was the start of 15 years in the lab, so I spent half my career doing lab work, half it doing public health work. And Piedmont Hospital was quite a small hospital on the edge of Dublin. And a lot of people told me, don't actually go there, it's a bad career move as your first job. But I chose to ignore it, which a lot of people here seem to be ignoring advice as well, so why not? But I'm glad I did, to be honest with you. I'm glad I went. Now, admittedly, the advice was don't necessarily do it as a long-term career move, but it was portrayed in a way that, well, maybe you shouldn't do it at all. Maybe stay where you are, you know, stay in the one place you were trained in. But I had a fear that if I stayed in the place I trained in and kept on going up the ranks, I'd always be viewed as a student who stayed on. So I wanted to go away and actually do something somewhere else, get a bit more experience. So because this was a small hospital, it was multidisciplinary. So I had to rotate between the different departments, which is quite useful. Because when you move to certain hospitals, if you're on call, you may actually have to cover blood bank or hematology or biochemistry all at the same time as your own discipline, or your own preferred subject, sorry. So that was just about a year, and the only reason I left is because I had to. They were bringing in cutbacks, they were going to let one person go, and last in, first out is usually the way it goes. It was either going to be me or someone else who had a family and a mortgage, so I chose to go. So I went back to college and did the diploma, which was essentially a 1 year course that used to be split into 2 6-month blocks with a year's in service in between. But because there was a shortage of jobs, they had to rearrange the course, and they allowed you to do the 2 6-month blocks back to back. So everything was out of the way in one sweep. So that was quite handy. So after that, I changed tack in my career again, and I went to ICP in Dublin, which doesn't exist anymore, but it's nothing to do with me. I left long after it went down the tubes. But that was actually a clinical trials place, and I always was interested in micro. And what I ended up doing in the diploma was specialising in micro. I presume you've read it ahead, even though I didn't mention it a minute ago. And I used to do micro for phase 2 and 3 clinical drug trials. Basically, they were testing new antibiotics. I had to do the usual kind of sensitivity tests, using the same kind of organisms and so forth, and using FDA type regulation, as one of the other speakers mentioned. And this was a precursor to accreditation. A lot of places didn't use these kind of rigid standardized techniques, but we were FDA approved as well. It was an unusual experience, but one of the advantages of actually moving sideways like that is I saw the non-health side of things. This was like a private company. It wasn't in the health service, and it was still a relevant discipline, very similar work, but it actually exposed me to the LabStar computer system, which is an older system by today's standards, but that actually landed me the job in Galway further on. So the reason I'm even mentioning that is sometimes a sideways move may seem like a daft move or a bit risky, but actually some of the things you pick up along the way will actually help you out further on, and it may not always be apparent what direction you're going in at the start. So after that, I actually went back to James's full-time employment, because I was worried that I wouldn't actually see enough of the stuff that I'd need to see to get through my exams, going forward. So I thought, no, I need to get out of the research side of things. And I went back, got lots of experience in very specialised areas in James's, and again, that actually helped me go further. I then was faced with a dilemma, because I had a 6-month contract being offered. I was on a succession of short-term contracts, maybe a month here, 6 months there, or whatever. Eventually you get cheesed off. As you move on in life, you actually want to have an anchor, somewhere to actually put down your roots or whatever. I thought, well, this is getting a bit much, maybe I should look elsewhere. And I also was afraid of being pigeonholed as being the student who stayed on again. So I thought I'd chance my arm with this. I had a friend who actually mentioned that there was a possible job going in GUH, and I just chanced the phone call, was talking to the chief, and everything was hunky-dory. He seemed to actually want the experience I had. But the clincher was the previous computer experience. It was the very system they were implementing. That's why they were recruiting. So as I say, sometimes a sideways move has benefits further on. So that brought me to Galway. So I was there for 10 years, so I would have met some of your predecessors, students going through, and to be honest with you, they've always been brilliant. And I'm not just saying that because I'm here, in case I don't get out of the car park. And similar to the presentations that have gone ahead, very high quality of students, really, really knowledgeable, but still always ask really good questions when they're in the lab. So while I was in GUH, I went on again. I went and did a MSc in Medical Micro in Bristol. And the reason I did that was there weren't too many options for MScs here. There was Coleraine as one option, which some people went through, but I actually preferred to go to Bristol, partly because my folks are in Dublin. I could fly from Dublin and see them on the way home or on the way out. So stupid idea, but it actually worked out well for me anyway. So after that, I got sick of the lab, basically, as some of my previous speakers have actually done in a shorter time period, I might point out. And that's actually why I asked yourself, why did you miss the lab work? I'd actually got to a point after 15 years where I didn't really think I was going to see anything more interesting. Like at a certain point in micro, you'll see so many of the unusual bugs that you think, well, what am I going to see after this that's going to be new? Now, admittedly, every job becomes routine after a while, so that would creep in anywhere. But I'd seen this type of job advertised in around 1999. Now, there's a national surveillance centre. It's now called the Health Protection Surveillance Centre, based in Dublin. You may or may not have heard about it. I believe a little birdie told me earlier that people were being told about notifiable diseases, so I might ask questions in a minute and see if you were listening. Not to me, but to your lecturer. But in around 1998, HPSC was set up as the National Disease Surveillance Centre with those initials. I don't know why they rebranded, but this happens a lot, same with the health boards becoming the HSE. There's no identifiable change in what they're actually doing, but they decided a name change actually makes things better. So they were started around 1998, started recruiting surveillance people in 1999. They started off with 2, and the idea at the start was that they'd use people who came up through the medical laboratory science, as it was called at the time, or medical science now, that pathway, with a special interest primarily in microbiology. Now, not exclusively that, because you would have done some of it, leading up to your degree level, so you'd probably get in anyway. But there was a divergence away from the original plan, so now there's a mixture of people from various different backgrounds. At this stage, I'd say there's 3 out of maybe 11 or 12 public health-based surveillance scientists who have my kind of background, have come up through the lab system. Whether they did PhDs, MScs, or whatever is irrelevant, but they all came up through a similar kind of pathway. But a lot of the other graduates would have done other science degrees or had research experience in the likes of Chagas, as someone else mentioned. What did I want to say after that? You'll have to forgive me, I'm a bit older, so I'm inclined to forget what I'm actually going to talk about next. I'll just have a look at my cog sheet here, which all people going to exams should have with them. Oh yeah. So essentially there's 1 or more surveillance scientists in each public health department. Now, each of the old health board areas is essentially a HSE area. Now, again, it's back to rebranding. It didn't change anything. We still do the same job, work in the same areas. And it's not standardized the way things are done. As in, people are recruited on different salary scales. They come from different backgrounds, so they don't share the same kind of knowledge, background knowledge, which in some ways is good for the likes of me, because I understand a lot more of this kind of stuff. I came from that background, so I understand what the lab results mean, whereas people from a pure science thing, they have major headaches trying to get their head around the lab results and what they actually mean. So now it's your turn. So it's audience participation time. Who knows what a notifiable infectious disease is? Don't be shy. I might ask for a volunteer. Or a point. Okay, I'll make it easy for you. A notifiable infectious disease is one of a scheduled list of diseases that are obliged to be notified to a medical officer of health, is the actual term. And basically any clinician, I'm going to answer the 3 bits all in the one go. Any clinician who actually diagnoses one of these listed diseases is obliged to notify the medical officer of health. And that person is actually a doctor. Medical officer of health is a function held by either director of public health or his nominee, and they can only nominate down 1 level. So it'll be a specialist in medicine or the director. There's 1 of those per health service area. So you can do the maths. There's less than 12 in the country. So who notifies? Any clinician who diagnoses one of those conditions. Now that's a GP, hospital consultant, or both. And there's also an obligation on a clinical lab director. And essentially it's usually not the actual director of the lab themselves that notifies, it's a nominee. And that typically is a lab-based surveillance scientist. So there's 2 aspects of this job. Mine is public health based. I work in a non-lab environment, which our friend over here who didn't like actually lab work would probably appreciate. It's a desk job. And my counterpart in the laboratory does both sides. So she feeds information over towards me. I willingly accept it, and I don't have to get my hands dirty in the lab, which is kind of handy in some ways. Why do people notify? Well, A, it's a legal obligation. So then you have to ask, well, why is there a legal obligation for these things to be notified? A lot of diseases, when you make a diagnosis, you're actually addressing the patient. You're actually finding out what's wrong with them, can we treat them, and how best to proceed. With some diseases, as in the ones listed, there are actually ramifications for the general public as well. Let's say someone is diagnosed with Ebola and no one thinks to tell anyone else. What do you think happens? We wouldn't all be here today. Half of us would be gone. And that's the most extreme version. It hit the papers relatively recently, and it's very much in people's mindset. They think that's the killer bug, that's the big problem. But even things as innocuously sounding as mumps or measles, they have ramifications as well. People can die of either of those. That's apart from other complications like brain injuries, orchitis, which is very unpleasant if you're a male. It involves swelling of the gonads. I won't go any further with that, and it can lead to sterility. And measles is incredibly infectious as well. Part of the reason it's on the list. If I walk into a creche as a toddler and I have measles, and there are 10 people in that room with me, 9 of those people are going to go away with measles and go on to infect and infect and infect. And before the introduction of the measles vaccine, it was a killer. In the year 2000, because of a fall off in vaccination rates after a paper that was published in 1998 by Andrew Wakefield, there was a lack of interest, well, that's the wrong phrase, there was actually a lack of trust in the MMR vaccine, and it was completely biased. It wasn't a very honest paper. It should never have been accepted for publication, but it did huge damage. And in the year 2000, 2 years after that publication, because of the public outcry and the lack of trust of the vaccine, 2 kids died in an outbreak in Dublin. There was about 1,800 people involved, hundreds of those hospitalised. I don't even know how many ended up with more serious complications, like SSPE, subacute sclerosing panencephalitis is one of the complications. Pretty nasty. It doesn't happen that frequently, but it's a very unpleasant complication. So some of the diseases we tend to roll off the tongue, like measles and mumps being, you get swollen cheeks, with, chipmunk features with the mumps. That's a fair description for most people, but it isn't for everyone. And it was the most common cause of viral meningitis until the introduction of the MMR vaccine. It's rarely a cause of viral meningitis now. We've had 1 or 2 in the last 5 years. It's so rare. And we've had massive outbreaks of mumps in the last few years. So it just goes to show the effect of vaccination on a decrease in disease rates. So that more or less covers the why notify. But the other aspect is that some of these diseases, like Ebola, the more epidemic proportion diseases, smallpox, anthrax, the bioterror kind of agents, they will always be on a list of notifiable diseases in every country that's a signatory to the WHO, because they have such global significance. And we have to make immediate notifications to WHO of certain diseases. Like cholera be one, obviously Ebola be another one, smallpox, polio, those kind of ones. It requires immediate action because it can sweep across the planet very quickly. Oh, sorry, forgot to press the button. So what is this? I don't know if that's in focus for everyone out there, I can barely see it from here. But it's actually the list of notifiable diseases. And we're going to spend about 2 hours now going through each and every one of them in detail. I'm sure you'll be glad to know that. Basically, I'll shortcut it in case anyone wants to count them. There's 83 listed. Some of them you will see zeros in, they won't actually be reported, but we still have to be aware of their presence. The likes of anthrax will be up in the top left. Most recent addition is on the bottom right in Zika. No surprise there because it's of global concern and interest. And we also have the more exotic ones, the smallpox, and Ebola is in amongst the viral hemorrhagic fevers there towards the bottom. So that's the full list. Most of those we actually get cases of, but there's a few that hopefully we'll never see cases of. But just because we won't, it doesn't mean we shouldn't be prepared. So people actually are always producing protocols to actually deal with and contain and share information on potential cases of Ebola, potential cases of smallpox and so forth. So there's a readiness there, it has to be there. We have an isolation facility based in the Mater Hospital for people with Ebola. You will have seen very unfortunate pictures maybe during the Ebola, I was going to say pandemic, the Ebola scare in West Africa, where an exercise in Dublin was actually being photographed, and there were some very unfortunate pictures in it showing things actually not going too well. You had a guard with no protective gear on standing beside a guy in full bodysuit, which didn't look right to me or any of my colleagues, but actually probably looked fairly okay to someone else. It was kind of a scary picture to have, but it was an exercise. It wasn't actually the end of the world anyway. What else would we have there? Nothing else. Let's skip on to the next one. And you can ask questions if you want. I don't mind you interrupt me as we're going along. I'd rather you do that because I appreciate this 30 years of stuff here I'm trying to cram into a few minutes, and I don't really know what to focus on because the job I do now is relatively rare in terms of there's only about 12 of us doing it in the country. There may be more opportunities coming up anyway, and I'd rather you ask me whatever you want to know rather than me telling you what I think you might like to know. So what is my role? So I'm basically involved in the surveillance of notifiable infectious diseases for our 3 counties in HSE West. Now what's in brackets there is CIDR, and that stands for Computerised Infectious Disease Register. We use a computerised system for actually notifying our diseases. Now the legislation actually doesn't specify we use this. It actually specifies something with a slower turnaround time. It actually goes back to the old days of paper notifications, where a clinician would send in a paper slip into their public health department, which is where I work. And every week we would have an obligation to make returns to the national body, formerly NDSC, now HPSC. And that's a very slow system, as you can imagine. For something like Ebola, you don't want to be waiting for the postman to deliver something so you know what the figures are down the country, because half the country will be gone by the time you actually find out what's going on. So in 2004 they piloted this system. It was custom built in Ireland, or not in Ireland, for our very specific set of parameters. We wanted to tailor it to the diseases we had. We wanted to be able to add extra information. Like your core notification is essentially like a credit card slip, essentially, if you look at the paper version. It's got the name and address of the patient, date of birth, where they might live. Oh, sorry, that's the address, obviously. Clinician, what the disease is, crucially enough. Sometimes when the paper ones come in, the doctor has forgotten to put what disease the person has, which is not very helpful, so they usually get a phone call back. The date of onset, and various basic things like that. And there's a subset of diseases on that list where we are required to gather extra information for our National Surveillance Centre. And things like cryptosporidium, we'd have to gather information on maybe the water supply, what risk factors they may have, do they live on a farm, have they visited an open farm, pet farm, or anything like that in the recent past where they might have picked up the infection, whether they've been to a swimming pool. Some of these are related to whether they've actually acquired it somewhere or whether they may pass it on somewhere. We would also be asking details about their parents' occupations. Are they healthcare workers, where they could pass it on to someone who might have a compromised immune system? Do they work in the food industry, where they could pass it on to someone on their plate if they're not washing their hands or whatever? So there's a lot of extra stuff we gather on certain diseases. That's a small subset of what we gather information on. The TB data is humongous, like there's 72 questions I think on the TB form, so I'm not even going to start listing those. Crypto was the one that sprang to mind, so I thought I'd start with that one. So what do we do when we talk about surveillance? I'm basically looking for trends in infectious diseases or trends in notifications. So you could start with bare numbers and look, have we got an increase in this disease versus that disease for the same period last year or over the successive years before that? And it can be very simple. It can be based on memory, as in you might remember getting the case of such and such a disease last week from the same area. You think, well, potential outbreak here, whatever. And that's essentially what I'm supposed to be doing. I'm supposed to flag potential outbreaks or potential problems and take it to the medical staff. And we decide then whether there's a need to create an outbreak or have an outbreak meeting to discuss further, get more test results. Not necessarily test results on humans. It could be environmental test results, like water test results and so forth. So that's the way it cascades. You start off monitoring, then you go on to flag things to the people who actually would be taking the action. And it'll probably make more sense when I get onto a few slides that I have for you that will actually show the various things that would have been investigated. So I would serve as a member of an outbreak control team. Essentially that's a multidisciplinary team meeting which would involve medical staff within the public health department, the likes of myself. They would often be consultant microbiologists. And depending on the disease, you may have veterinary people involved, you might have the county council. A lot of the time we have meetings with Irish Water or the relevant county council because there's often a water component. Things like crypto would be a great example of that. And we'd add on extra members depending on what was actually going on. For things that might cause panic, we may actually have the emergency services involved, the guards may be involved. There could be public order implications of making an announcement about certain diseases. So there's a lot of disaster planning goes into this as well. It's not just a react straight away. There can be multiple different agencies involved and it has to be cascaded properly. And everything is confidential. If there was a leak of something that had potential to cause panic in the public, that would not be in anyone's interest. So everything is kept mum until it's released in a controlled fashion. That's why every time there's an outbreak, you get a statement released. One. We've had incidents in the past where there have been multiple little leaks, mini statements, for whatever political reasons, and they don't help anyone. It causes confusion because they often have conflicting information in them. Anyway, that's more about the workings of the public health department rather than my own role, so apologies for digressing. So what would my role be on the outbreak control team? So it's the next thing. It's essentially data analysis. We often have cases where I will bring something to the team and I would show them the information I've gathered, like the trends in such and such a disease. I might have maybe seen an increase in cases over a period of weeks in a particular area, in a particular age group, gender, what else? Ethnicity on rare occasions. I'll take it on and we'll see if anything needs to be done. It can also go in a different direction, where we get a call from Irish Water, say, and their routine monitoring of the water supplies has revealed crypto in the water. So this is in the water, as opposed to in a human. They would often contact me and see, not directly, via the Director of Public Health, to pull out figures for that area over the recent past or over the recent few years, and see if there's anything odd happening now versus the past. So I'd have to look at the more recent figures relative to the recent crypto positive in the water, and see if it's any higher or any lower than previous years. And it can be the basis for a boil water notice being put on. And I'd say most people here would have been subject to a boil water notice or lived in a boil water notice at some stage in the last few years. They're quite common. And sometimes it's due to treatment failures, sometimes due to a mechanical breach in a pipe. There's always people with JCBs going around digging things up, and they never know what's under the ground until they actually stick the bucket into it. So that's part of the data analysis aspect. So one is from first principles, where I actually see something odd and I flag it. Another is I'm actually asked for information about something that they suspect might be going on before it may even have gone on. So I would reiterate or confirm that there's a problem. And there's also a data analysis aspect of, let's say, a foodborne outbreak. We would often get, I was going to say complaints, but allegations that people who attended a function, maybe a wedding or a graduation or something similar, hopefully none of you will be affected by a graduation. Well, you know where I am, I'm just in Maryland Park here. We'd be told maybe about 10 people out of 20 who attended a function came down with the same kind of symptoms within X amount of hours out. So I might have to go and do some data analysis on that, maybe based on the actual menu that had been served. How many people had what out of the menu, and if they did have that, how many portions did they have? Did they have water? Did they have wine? Whatever. And we try and work out the most likely food item based on risk ratios. Very simple calculations, not high-end maths at all. So that would be 3 minor things that I would do in terms of data analysis for outbreak investigations. I'd also be a lab liaison person. Like, I would link with my counterpart in the laboratory here in Galway. If anyone's been trained in Galway in micro, you might have met Belinda. And there's also another person up in Mayo, Magella, and there's another one in Portiuncula, Michelle. So they'd be the 3 local labs that I'd be linking with. But I would also link with reference labs. We have a Salmonella reference lab here in Galway. There's a Cherry Orchard reference lab for VTEC, toxigenic E. coli. We have a TB reference lab in James's. The Meningo reference lab is in Children's University Hospital in Temple Street. And a lot of that will be to determine whether or not we do or don't have an outbreak. Like, having X amount of cases doesn't mean there's an outbreak of that disease. You also need to see, is it the exact same type of the organism? Like, if you have 5 salmonellas in the space of a week from a small town, it probably is an outbreak. But if they all turn out to be different types of salmonella, then it more than likely isn't. It could be coincidence. Maybe they're on holidays in Spain or Portugal or wherever, all came back with a dose of the you-know-whats. And then the final part that I put down here was liaison with HPSC. They're the national body for surveillance. They would often come to me for clarification on various things we'd actually have notified. At the bottom of the list of notifiable diseases I gave you, there's actually a little thing that you probably wouldn't have been able to see. It says refer to case definitions for further. Each of the diseases has different categories for notification. You could have a confirmed case, probable case, or possible case. There are slight variations. Sometimes there is no possible case or probable case. And they usually follow the format. A possible case is when a doctor diagnoses something or thinks that someone has something. That's a possible notification, or a possible case, and is a clinical notification. We also have a confirmed case, which is a clinical case that has been confirmed by a lab test result. Where my role would be quite useful in that is I also would know, well, does that result correlate with a confirmation, a true confirmation? Because if you have someone who, let's say, had been vaccinated with MMR, and they're tested for measles, and they have IgM positive to measles, well, that could indicate either active infection or prior vaccination with MMR. So that wouldn't necessarily be conclusive. So I would be looking more for a molecular result, where you actually look for the genetic material of the living organism rather than a result that might indicate past exposure or past immunisation. So that's probably enough on that anyway, before I go waffling on anymore. I tend to sidetrack myself. So this is an example of the kind of thing that I'd be doing. So I'd be up all night now with the crayons drawing this kind of rubbish. And this essentially came from a dump from the national computer system of a particular disease. I'll tell you what the disease is in a minute. and I basically take it into Excel. And on the bottom, you see all these numbers, 1 to 52, just in case you can't see what's written there from the back. That's basically the week numbers in the year. All the coloured bars at the bottom represent the different years. So this is one disease over the course of the weeks of the year, by year, and the number on the vertical column is the actual number of cases we had in that year. What I'd like to point out first is, from here around week 13 here, over to week 26, that's essentially quarter 2 of the year, which is spring. Sorry, my eyes are getting a bit sore. So that's spring, and what I want you to show you, ignore the big blue peak for the moment. There's a clustering there. You can see there's definitely a hump in virtually all of the actual traces going across there during spring. And that's normal, essentially, and that's what I'm supposed to do, is see what's normal versus what's abnormal. And this is a very simplistic view of it, and it also is intended to show you, well, that thing sticking up there is obviously abnormal, okay? But what my job is I have to see that before that happens. And if I tell you that that was cryptosporidium, would that surprise you? That was 2007. Okay, so a lot of you would have been living in here or visited here or had to stop drinking the water here because of that happening. And actually, what happened with this is, you'll see here, it starts over here, it may not be clear from the back. There's a little hump there, which is a bit unusual. And that essentially was little clusters of cases out in outlying rural areas. A little bit earlier than normal. Not that abnormal though, because crypto can be got from animals. It's one of these things called a zoonosis. It's not only from animals, because there's 2 different strains that actually predominate. One is hominis and one is parvum. Parvum can be in humans or animals, so that's the one you'd most likely get from the animals. But hominis is not carried by animals. And this outbreak was caused by a mixture of the 2, mainly hominis. So basically, when we were getting these ones here, we didn't know what type they were anyway. But because they were in rural areas and there didn't seem to be any obvious links to a common factor, they all lived on farms or close to farms, it was assumed that this was related to that. And it probably was, because that's what most of these ones here in the spring peak are. It's to do with calving and lambing. Young animals are more susceptible to the infection. They excrete it in large numbers in their feces. Ends up in the water systems or on people's Wellington boots, into farmhouses, 2-year-old crawling around the ground, fingers in the mouth. Classic. Well, not so classic for the kid though. But it can actually leave them very debilitated. I mean, it's an illness that can actually take 2 weeks to clear, and it's quite unpleasant, as anyone who actually got it would know. But this peak here was actually spotted in around week 11. We had boil water notice on in around week 11. So it was this segment going up that was noticed that we were getting cases that weren't just in the rural areas. Sorry, I left my teeth at home. My teeth tripped me up there now, chewing them. We noticed these people weren't all from rural areas. Maybe I should say they live in the country. Easier. We were getting a lot of urban area people who had no contact with animals, no other risk factors. They hadn't been to swimming pools. Initially, with some of these initial cases down here, we had actually noticed that they'd been in swimming pools as well. So we thought, well, it could be one person who has a fecal accident in a pool, contaminates the pool, and it's actually surprisingly common. Some people actually do it deliberately on holidays. Get a free holiday. Yeah, good, you know, it's called logging, apparently. Anyway, I digress. Sorry, I've distracted myself again. No, definitely not. No, it's nothing to do with me. Meanwhile, back at the chart. At the bottom end of that leg there that's going up, we did notice some people had been to swimming pools, but they were never the same swimming pool, which doesn't prove or disprove anything. It's just an additional risk factor. But by the time we reached that little shoulder there, it was starting to be more obvious we were getting cases with no other obvious risk factors or exposures. At that point, we were getting worried. So we had our outbreak meeting, we had environmental health involved as well, and we were in with the council. And it was decided in the interest of public health, public safety, we should consider a boil water notice, even as a precautionary measure. So this is around week 11 going into week 12. It's just before Paddy's Day in 2007. 2007. We're going all Michael Noonan, leaving out a 0. But it was another week to 2 weeks before we actually got confirmation that it was in the water. And for those 2 weeks, there was a lot of disbelief that this was actually going on at all. Now, can anyone explain to me why that peak went up when we put on the boil water notice there so early?
Because the cases maybe were just still coming in?
There's a small bit of that, yeah. The actual reason is the incubation period for crypto is about 2 weeks. So we put it in there. So people who might have actually drunk the water for the previous 2 weeks hadn't actually presented with symptoms yet. And that doesn't include people who passed it to their family members after that as well. So that will actually propel the spike upwards. So you can imagine where that spike would be if that boil water notice had gone on later. So we ended up with over 300 cases that year, and normally we'd have anywhere between 80 and 120 cases in a full year. And we had most of them in that period in March. And the boil water notice extended from essentially that shoulder there all the way over to, I think it was week 32. After that, it was all fine. It required a complete changeover of the water treatment plant. It was long overdue. Anyway, that's what I just want to say about that. But that's probably the most visual example of actually what I do or what I'm trying to do. And that was.
Do you set the threshold yourself, or does someone tell you that the threshold you should be aware of?
Which threshold? Sorry.
Say, for example, when it got to the shoulder, right?
Yeah.
Was it at that point that you knew to react?
No, actually, no. There's continuous monitoring all the way along. It's one of the diseases we do what's called enhanced surveillance on, and we look for people's individual risk factors beyond the core data of their name, age, address, and all that. So by actually looking through all the feedback we got, it was gradually becoming apparent that we were getting people who didn't have any other risk factors other than water. So there's no threshold as such. It was just that little shoulder there kind of corresponded with when we were starting to see an increase that was mainly due to people in urban Galway areas who had no contact with animals, couldn't possibly have got it anywhere else other than the water, or else there was something major going weird.
What's the peak there around week 15, 16, 17?
Sorry?
See the second peak, smaller peak, around week 17.
Week 15, 16, 17?
Yeah. Was that boil water still on there? Or was it boil water?
Oh no, that it continued on. The boil water notes was still in place, but you also have to factor in the fact that there'd be people who'd be getting sick, you know, going up in that spike, who can infect other members of their household. They'll present later and later. And there's also an increased awareness when there's a boil water notice like that on. People will go for testing more readily. Like, how many people here go to the GP if they get a dose of the trots? You don't tend to go in a hurry. What you tend to see, and what was happening at the early stages, is it's very young children are brought to the GP because there's more concern about a very young person or someone who has an immunocompromised condition, or someone who's much older, who will be more susceptible to dehydration. So we were starting to see younger people at the start, and then it was affecting all age groups. So that double peak isn't actually something actually going wrong. Again, it's more, it's like a reflection of what went up, but on a smaller scale, and it usually is family transmission. And that's probably only a snapshot of what was going on, because these are confirmed cases. The actual number of people who might have had diarrheal illness is probably way higher. There have been estimates that it's probably 10 times what we actually got. So you've got 2,000 to 3,000 people, which is quite substantial. So 70,000 people essentially were discommoded in terms of their water supply to avoid 70,000 people ending up with that. So that's an example of when it actually is working right. It's quite hard to actually, sorry?
I'm just conscious of the time.
Sorry, apologies.
Sorry.
I will scoot on. I'll scoot on. There's only one or two more.
We'll
Yeah, I'm just going to skim through this. Okay. H1N1. That's fine. No, that's great. I'll wrap up now in a second. H1N1, swine flu. That blue peak there towards, you don't have to read this, that's just a succession of flu isolates over time, going from 04 to 16. That's the pandemic flu. And you can see, because there's a vaccination campaign, you don't see much of that colour further on until you get towards this year. That's the predominant colour this year. We had a few hundred cases this year. People would have got the vaccine and have knocked it out then for quite a few years, and it's starting to come back again. That's all I wanted to show you on that. WHO actually formulated a vaccine for flu based on this kind of stuff. They would see what's circulating in the hemisphere. It's like the flu season is split into two hemispheres in the world. Its monitoring is in the winter period for both halves, and they formulate the vaccine for the next season based on this kind of data. What's the predominant circulating strains? That's all I wanted to show you on that. This one is, what is this? Measles. You can easily spot the increase in cases in the middle, okay? There was a succession of outbreaks in both of those years in the middle. But what I want to show you is it's not just pure numbers and something appearing as a spike. On the extreme right, you'd see a very small bar in 2016. That's actually an outbreak of 2 people. And the only reason we know that is because they took place in a very short space of time with each other. One person came in and infected another person. So that's actually an outbreak over there. So it's not always big spikes in the sky. Okay. I believe you've been talking about meningococcal vaccine group B to some students. And can anyone explain to me why meningococcal group C is actually so low on this? It's the same format as before, years gone across the bottom, numbers of cases up here, and group C meningo is very, very low there. Okay, I'll short circuit it. Essentially, group C meningo vaccine came in 2000. That's why it's so low. Before 2000, you'd have the same kind of spikes in the group C column as you do in the group B column. And hopefully when the B one comes in, that'll go down to those levels. But what tends to happen is a different group shoots up as the predominant group. But hopefully the numbers will be lower. Again, let's scoot on. Now, if anyone wants more information, I'd certainly recommend you have a look at the HPSC website. There's lots of stuff on there. Also, in your in-service training year, if you're in any of the larger hospitals, you will probably meet one of the surveillance scientists in the lab. They will be able to tell you what they do, because it differs slightly to what I do. There's links between us, but they're not the same function. The other thing I was asked to do was give advice to my younger self. I wrote it on the page, but I forgot to put it in the slide. Essentially, my path to being here has been very kind of twisty, turny, all over the shop. And I would say don't be afraid to make a choice that actually takes you in a different direction to what you might have originally thought you'd go in. Because it's all experience. You can't lose. And you can always go backwards again or change direction again. And the only other piece of advice I'd say is be good to the students. Remember what it was like when you were going through. I still remember. And I'd like to encourage people the same way as I was encouraged. Some people don't want to engage, but don't be those people. Do try and help your fellow students when they're coming through. Assuming you actually want to work in a hospital. And that's me finished. Sorry, Mary.
Has anybody got any specific questions for Rich? Stephanie?
The list of diseases, is that set in stone for Ireland only, or is there a link between different countries?
That's our one.
And does that, say if for example, leave here, France or somewhere, would add something on, would we be triggered to update?
Not necessarily. We tend to mirror what the WHO wants or is interested in, with some variation. And no two countries will have the same list exactly. There are notifiable disease lists all across Europe, and they may vary. Like in Scotland, there's scarlet fever is notifiable. That's not notifiable here. By and large, they do follow the same kind of format. But that list has been amended multiple times. It's specified in the Health Acts from the 40s, and it can be amended as needed. Swine flu was added, instantly. Zika was added instantly as well.
Do we ever remove any diseases?
Loads were removed. There was a lot put on in the 80s. There's one that always puzzled me. It was before my time anyway. Candida used to be notifiable in the 80s. And you think, why?
I think, fourth years, you probably should relate a lot of this with your epidemiology module. For some of the others, for you going on placement, surveillance scientist role, it's good to be aware of it. But certainly, the third years, we will talk about vaccination on Monday, and certainly Debbie with the micro. So it's really important to see the multidisciplinary approach here for Rich's role. And we will wrap it up now if that's okay. Yeah. Unless anybody has something specific or anything, there is hopefully still tea and coffee in the foyer.
I'm sorry if there isn't.
No, it's our own fault. I booked it too early. It was my fault. But if there's the speakers are going to hang around if there's anything that you want to ask. But thank you very much for coming. On a separate note, guys, hang on. Completely separate note. Okay, I'm using the opportunity that you're all here. First of all, to say thank you to all of you that took part in the survey that I have circulated. First years, anybody that's here, if you haven't had a chance, lowest year for response rate, so maybe you could bring up the first year